These are the lecture notes for Chapter 16. It's called The Molecular Basis of Inheritance, but it's actually about DNA. So here we have an image of the structure of DNA, and you probably recognize it because you see it, all the time in all different places. But basically, it's a double helix, which is double helix is the name for a twisted ladder. And the structure was proposed in April 1953 by James Watson and Francis Crick. And they actually received the Nobel Prize for their work. The way that DNA works, I know everybody knows, or most people know, at least by the time we get this old, we know that DNA codes for our traits. It has a code that codes for our traits and kind of makes us who we are, makes us look the way that we look, and even somewhat act the way we act, and maybe even makes us, susceptible to certain diseases and things like that. But what DNA really is the DNA code is broken up into pieces called genes. So a gene is a sequence of DNA that codes for a particular protein. Okay, so what you're seeing here is basically the DNA before it replicates, and then the DNA replication process. Basically, the original DNA strand, which we call the parent strand, unwinds and unzips, and a new strand is added to an old strand. So since DNA has two strands, each of the two old strands is used as a template to copy a new strand. And this is what happens when we go from a single-stranded chromosome before the S phase of interphase, and then it becomes a double-stranded chromosome with two sister chromatids. So DNA replication happens before cells divide. That's one of the things we'll study in this chapter, and also we'll study how DNA codes for protein. So we're going to do a little bit of history. There was a time before we even knew what DNA was, and then Gregor Mendel discovered his laws of heredity before we even realized that DNA is the molecule that codes for our traits. So sometime in the early 1900s, I know it was in the 1900s, scientists began looking at, we knew that chromosomes consisted of DNA and protein, but we didn't know if it was the DNA that contained our genetic code or if it was protein that contained our genetic code. So there were several experiments that were done that gave us the information that we needed to know that it was DNA. So we start with Frederick Griffith in 1928. Frederick Griffith was working with two strains of bacteria. He wasn't really studying DNA at all. But in 1928, Frederick Griffith was studying two strains of bacteria. One, it says pathogenic. Basically, it was deadly to the mice that he was infecting with those bacteria. So there was a harmless strain and there was a pathogenic or deadly strain. And what happened is he tried different things in the laboratory in a test tube. He ended up, he knew that when he injected the pathogenic strain into mice, they died. And when he injected the harmless strain into mice, they didn't die. So what he ended up doing is he heat killed the pathogenic strain. So if he injects heat killed pathogenic strain into mice, they don't die. But he put the heat killed pathogenic bacteria in a test tube with the harmless bacteria. Then he injected that into mice, and they died. So what he discovered was this phenomenon of transformation. So something from the pathogenic bacteria, even after they were dead, was ending up in the harmless strain of bacteria and was now causing them to be pathogenic and deadly to mice. All right, we know now that was DNA, that DNA was entering the harmless bacteria from their environment, which in their environment, he added the heat-killed pathogenic bacteria. So the DNA from the dead bacteria was entering the live bacteria, and they were expressing that DNA. They were expressing the deadliness of it. They were making the mice sick and they were dying. So here's an actual illustration showing you the experiment. When Griffith injected living S cells, living S bacteria, the S is the deadly strain. When he injects living S cells into mice, they die. Living R cells, which is the harmless, non-pathogenic strain, the living R cells, mice don't die. When he heat killed the S cells, so now they're dead, but when they were alive, they were deadly. So when he heat killed the S cells and injected them into the mice, it didn't kill them. But when he mixed the heat killed S and the living R, which by themselves did not kill mice, when he mixed them together in a test tube, let them grow for a little bit, injected them into the mouse, it caused the mouse to die. All right, and that is what Frederick Griffith is given credit for, is discovering the process of transformation. Transformation is when bacteria take up DNA from their environment. In this case, the environment contained dead pathogenic bacteria. Okay, then there was more work done by Oswald Avery, Macklin McCarty, and Colin McLeod. And what we're doing here is gathering evidence for DNA being the genetic material and not protein. So, okay. I'm just wanting to make sure of the experiment that I'm describing here, because there's two that are on that are experiments on bacteriophages. I want to make sure I'm telling you about the right one. So, bacteriophages are viruses that only infect bacteria. So they're deadly only to bacteria. Now, a virus, the structure of a virus is DNA or sometimes RNA, but a virus is going to have a core. So in the core of the virus, you have the genetic code, basically. And then outside of that, there is a coat. Sometimes it's just protein. But it's basically viruses are just DNA and protein. DNA is, or RNA, but there's a genetic material on the inside and then a protein coating on the outside. But without knowing specifically which one contains the genetic code, scientists needed to prove it was DNA and not protein. So they studied, this is what a bacteriophage does, by the way. It kind of looks like a spaceship. The green one is the bacteriophage, and it looks like it's a spaceship kind of docked on the moon or something, or a planet. But the brown that looks like a planet is just the part of the outside surface of the bacterial cell that this virus is infecting. This green virus is called a bacteriophage. All right, inside of the core of the bacteriophage is DNA, and then this outside green part is the protein coat. So what we have learned through several different experiments is that the bacteriophage protein coat does not enter the bacterial cell. What it does is it injects its DNA into the bacterial cell, and then the DNA codes for the production of new viruses. So it's got to be the DNA that contains the genetic code. there is an animation on slide 12 of the reproductive cycle of phage T2, which is the bacteriophage that infects E. coli bacteria, and that's the one that was being studied. So there was one experiment. There was some experiments that were done by Avery and McCarty and McLeod on these bacteriophages, and then we had an experiment in 1952, which is very close to when Watson and Crick discovered the structure of DNA. But Alfred Hershey and Martha Chase were studying this T2 phage or T2 phage, anyway, this bacteriophage that infects E. coli bacteria. And they were trying to discover if it was DNA or protein that contained the genetic code that told the bacterial cells to make new viruses. They knew that the bacteria E. coli was the host and that the virus needed a host cell to reproduce, but they did not know if it was DNA or protein that was responsible. So here's what they did. They radioactively labeled the protein with radioactive sulfur, and they radioactively labeled the DNA with radioactive phosphorus because the protein contains a lot of sulfur and the DNA contains a lot of phosphorus. So they could distinguish between the protein and the DNA based on the detecting the radioactive sulfur or phosphorus. Okay, so they put these bacteriophages and the E. coli bacteria, they put them into a flask and let them grow and become infected. And then they mixed the infected E. coli that was infected with the bacteriophage. They mixed it in a blender, and then they put the contents of the blender into a test tube and centrifuged it or spun it so that the heavy stuff fell to the bottom and formed a pellet, and then the lighter materials formed a liquid layer at the top. So what they found is that there was no radioactivity in the pellet, and that the radioactive sulfur showed up in what they call the supernatant, or the liquid above the pellet. In other words, the protein never entered the bacteria. Then they did another experiment where they used the radioactively labeled DNA, did the same thing, mixed viruses and bacteria, let them grow in a flask, and then put the flask in a blender, mixed everything all up in a blender, poured that into a test tube, centrifuged it so that the heavy stuff, the bacteria, would fall to the bottom, and no radioactive DNA showed up in the liquid at the top layer, but the pellet was now showing the radioactively labeled DNA in the pellet. So what that tells us is DNA enters bacteria, not protein. So it's got to be DNA that contains the genetic code. Now, there's a good animation on Hershey and Chase's experiment on slide 15. Okay, so we know now that DNA it's got to be the genetic material. We had the evidence from the transformation by Frederick Griffith when he showed that DNA, or when he showed that something is entering the non-pathogenic bacteria that is causing it to be pathogenic. And now we know that the substance is DNA because the only thing it could have been was DNA or protein. And we eliminated protein with the Hershey and Chase experiment. Okay, so that's where we are right now, but there is a little more evidence that led us to discover the structure of DNA and understand how it works and how it codes for our traits. So we know that we learned this in one of our first few chapters on chemistry. We learned that DNA is a long polymer. It's a long chain. And the monomers or building blocks are called nucleotides. And each nucleotide has a nitrogenous base, which we can just call a base, a sugar called deoxyribose, and a phosphate group. So basically, we know that DNA is two strands of nucleotides, two long strands of nucleotides. And we know that there's four bases, four possible bases. They are adenine, thymine, guanine, and cytosine. And that the different combination of these bases is what makes up our genetic code. All right, now we have in 1950, which is before the Hershey and Chase experiment, proving that DNA is actually the genetic material. But in 1950, Erwin Chargaff was studying the composition of DNA. He was a chemist. And what he discovered was that in any sample of DNA, it doesn't matter, any sample of DNA, and he used different species, like it didn't matter what species it was. In any sample of DNA, the number of adenines and thymines would be equal, and the number of guanines and cytosines would be equal. So it's called Chargaff's rules. And this is just an illustration of like a single strand of DNA. It's just showing you one strand. DNA really has two strands, but this is showing you like one nucleotide, one nucleotide would be this phosphate, sugar, and then the base thymine. So this that I'm highlighting is a nucleotide, okay? And the bases that you see here in the far right, thymine, guanine, cytosine, adenine, these are the ones that the different combinations of these bases determines our genetic code, our traits, right? So in the structure of DNA, this is an animation on DNA structure, and it is slide 19. The structure of DNA it's actually double-stranded, and there were several scientists that were working on building a structural model of DNA, and they were doing it in different ways. They were using the information from Chargaff, that the number of A's and T's were equal, the number of G's and C's were equal. And then Rosalind Franklin and Maurice Wilkins were using X-ray crystallography. And they weren't just studying DNA. This was just one of the things that they were doing. That they were using X-ray crystallography, which is a technique that can show you give you a visual of the shape of a molecule. And we didn't know the shape of DNA at the time. We didn't realize it was a double helix. So Rosalind Franklin took a picture of DNA, an X-ray crystallography, generated image. And I don't know about you, but I can't look at this image, this X shape here in this image, and tell that that is a double helix. But she could. Anybody that studies molecular structure would know that if you take a picture of the top, from the top of the DNA molecule, that this is what it would look like if it was a double helix structure. And I've never been able to see that when I look at this image, but I know that she could see it. And she reported that DNA is a double helix structure. So now Watson and Crick took her information, knowing that the DNA forms a double helix, Chargaff's information that A's equal T's and C's equal G's, and they determined that DNA must have a double helix structure. This animation of the structure of DNA is slide 25. And that A's pair with T's and G's pair with C's. So basically it works like this. So adenine always pairs with thymine, and adenine has 2 rings and thymine has 1. So that means that the steps or rungs of the DNA ladder are always composed of 1, 2, 3 rings. One of the bases is a purine, and it has 2 rings, and one is a pyrimidine, and it has 1 ring. So the steps of the DNA ladder are always composed of a purine and a pyrimidine, so that the steps are always 3 rings wide. They're the same width. Okay, so adenine always pairs with thymine, guanine always pairs with cytosine. And I'm trying to find a picture of the actual shape so you can see, like, not just one strand of it, but I can't. So I need to stop this recording and start a new one. So I'll just go back to, all right. So what Watson and Crick discovered was that DNA it's a ladder, and the sides of the ladder are composed of the sugars and the phosphates. The steps of the ladder, or the rungs of the ladder, are composed of A's paired with T's and C's paired with G's. So they're composed of base pairs, is how we say it. So that is basically a description of the structure of DNA, is that it is a double helix or twisted ladder where the sides of the ladder are the sugars and the phosphates, and the steps of the ladder or rungs of the ladder are the base pairs, A's and T's and C's and G's. So now we will start learning about DNA replication, and this is slide number 32. So we'll start with slide 32 in the next recording.